I'm Mark Chiu over at Springhouse, Pennsylvania, working with Janssen Pharmaceuticals, and the division is with Biotechnology Center of Excellence, and our group focuses on this theme of biologics engineering. I think it's one of the most exciting areas is trying to delve more deeply in the field of immune oncology. One of the great promises is trying to understand the balance of immune stimulation as well as immune checkpoint control. By using these two aspects, we can now be able to tune and ask more directed questions about how we can use a patient's capability to essentially control their disease state and disease tissues and cells. By doing this, you can use a lot of the host material and not worry so much about exogenous factors which may interfere with potential toxicities. One of the largest challenges in terms of the development of immune oncology targets lies in the difficulty of doing translatable experiments. Often when we do our work, we as biochemists or molecular biologists do a reductionist point of view of thinking, where we isolate a particular target and try to develop models with regards to the cells and put them into a defined animal model system. The problem of translating that is trying to understand the role of disease tissue distribution versus normal tissue distribution. And not knowing the interplay between these normal and disease tissue is often what we cannot do well in the preclinical models until we do some clinical trials. Being able to understand how we can do those kind of studies will significantly advance our field in the drug discovery effort. One of the great challenges in being able to understand how best to engineer molecules is to try to take into consideration aspects of the flexibility to have the induced fit of what you want to have. But with this flexibility, you have the increased risks of potential risks for manufacturability. Manufacturable molecules often like to rely upon more stable molecules, but having more stable molecules may not be flexible enough to have the biology that you want. Understanding the limits of these will be one of the more critical aspects of advancing these differently architectured molecules in the future. So when one goes beyond just a single monospecific antibody, which is bivalent, there's lots of interesting opportunities by which protein engineering can derive getting a bispecific molecule. Now, if you take the analogy of whack-a-mole in terms of trying to shut down particular targets, having the architecture by which you can shut these things down is going to be most critical. It's not going to be clear whether the interaction surface of a Fab arm of an antibody is going to be sufficient. Maybe one has to think about orienting the binding arms in different locations with different specificities, with different flexibilities, with different interchange and exchange of the domain movements that are intrinsic with a protein molecule. So when thinking of going from a monospecific to a bispecific to a multispecific, the ability of being able to control and understand how such modulations can affect functions is going to be most critical. Now, the aspect of trying to engineer more specificity is not going to be difficult. Rather, it's going to be trying to understand what its impact may be on the manufacturability and the regulatory aspects, and trying to understand the relative safety and efficacy, as well as what the profile of the toxicity may be.